topic is development of chick. Today is my second lecture. In the previous lecture, I have explained you about the fertilization and cleavage. Today we will study morula stage, blastula stage and the process of gastulation. What is morula stage? Morula stage is the stage in which morula is formed. What is morula? Because of the consequence of cleavage, there is number of blastomeres are formed, in the result of which mulberry-like structure is formed, in which the central cell is smaller and outer cell is larger. And these cells are compactly close with each other. This stage is termed as morula stage and the structure of embryo is termed as morula. This stage is very short stage and the embryo enters into another stage, that is blastula, by splitting the blastoderm into two layers. One is the epiblast and second one is the hypoblast. This is epiblast and this is hypoblast. In the result of which a fluid-filled cavity is formed, which is termed as blastocoele. And because of the formation of blastocoele, the stage is termed as blastula stage. The cell of the margin of the yolk is making junction with the yolk, which is termed as area opaca, while the central part of the blastoderm is termed as area pellucidum. At this stage, hence lay egg. When the egg is laid in the environment, no further development takes place in the embryo and embryo becomes inactive. After laying egg, when the temperature, that is 36 to 38, is provided to the egg, then the further development is started. The required temperature, that is 36 to 38 degrees centigrade, is provided to the egg, is termed as incubation period. Incubation period can be natural and artificial. Natural incubation period is the period which is given by the hen's mother, while artificial incubation period is the period in which 36 to 38 degrees centigrade temperature is provided to the embryo by keeping it inside the incubator. During incubation, the two processes take place. One is the gastulation, another one is the neuralization. The process gastulation takes place in the stage which is termed as gastula stage, in which cell move inward and in the result ectoderm, endoderm and mesoderm is formed because of the movement of cell to definite location. In all the apes, the cell that is provided to the embryo is by the epiblast. Epiblast gives cells for the further development of the embryo. The archetron that is formed because of the movement of cell toward the center is inner side. While this type of invasion is take place in the frog. The cell move inside, in the result of which blastopore is formed and a cavity start to form is termed as archentron. While in this type of gastulation process, archentron is formed without imagination, in which presumptive mesodrome and presumptive endoderm is formed because of the movement of cell toward the center of the blastodisc. When the cells start to migrate from the epiblast to the marginal line of the blastodisc, then a groove is formed. This groove is termed as primitive string. The cell then detach from the epiblast and move inward, in the result of which groove is present. This groove is termed as primitive node or Hansen's node. This node is formed at the anterior end of the primitive state. This is the swelling portion of the anterior of primitive state. This primitive state elongate because of the movement of more cell toward the center and movement of these cells inside the duct or disc. This invasion is functionally similar to the blast